Since its inception more than 3 decades ago, chromatin immunoprecipitation, or ChIP, has become the most widely used method for chromatin mapping. It offers powerful insights into the epigenetic mechanisms associated with human diseases, including many types of cancer. But its low resolution, high background, and expensive price tag makes ChIP less than ideal. In response, researchers have developed new immunotethering strategies, which are gaining widespread popularity in the chromatin field. The next advance in these strategies is CUT&Tag, a novel assay under Epicipher's CUTANA platform for ultra-sensitive genomic mapping approaches. CUT&Tag stands for cleavage under targets and tagmentation. In CUT&Tag, extracted nuclei are immobilized on a solid support and then treated with a primary and secondary antibody to label a histone post-translational modification, or PTM, or to label a chromatin-associated protein of interest. Next, the nuclei are treated with a fusion protein made of protein A, protein G, and a TN5 transposase preloaded with sequencing adapters. This PAG-TN5 is recruited to the antibody-bound chromatin and activated by the addition of magnesium. This catalyzes tagmentation, where the antibody-bound chromatin is simultaneously fragmented and tagged with sequencing adapters. Tagmented chromatin is then directly amplified by PCR and purified for next-generation sequencing analysis. CUT&Tag allows users to bypass standard sequencing library preparation, providing a streamlined, 2-day protocol that goes from cells to sequencing in a single reaction tube. In fact, published research papers have reported adapting CUT&Tag for single-cell studies, an application that has been challenging when using ChIP. Epicipher's optimized CUTANA CUT&Tag protocol allows investigators to generate high-quality mapping data using as few as 1,000 cells and 3 to 5 million sequencing reads. CUT&Tag is highly complementary to its sister technology, cut-and-run, which utilizes a slightly different fusion protein, a protein A-protein G hybrid fused to micrococcal nuclease to selectively cleave antibody-bound chromatin. Cut-and-run is a highly robust technology, extensively validated using a variety of cellular inputs, sample processing methods, and targets of interest. It generates high-quality data for a variety of targets, such as different classes of histone PTMs and diverse chromatin interacting proteins, including transiently interacting factors, which have been historically difficult to profile using ChIP. The newer CUT&Tag approach is optimized for histone PTMs and select chromatin-associated proteins. While CUT&Tag has incredible potential to transform chromatin research, its broad compatibility with diverse sample inputs and target classes is still in development. Together, Epicipher's CUTANA CUT&Tag and cut-and-run assays generate high-quality chromatin profiling data using fewer reads and cell inputs, and less hands-on time compared to ChIP. Epicipher has launched their CUTANA platform for CUT&Tag and cut-and-run assays, providing the immunotethering enzymes, validated antibodies, companion reagents, and optimized protocols required for a successful experiment. If you need guidance from choosing an assay or designing your experiment, please reach out.